Hello friends, welcome to Creative Medicine. In this lecture, we will learn about.
Hello friends, let us now learn some important points about the synthesis of pyrimidine. This pyrimidine synthesis occurs mainly in the liver. So here carbon dioxide combines with the compound glutamine, and in the presence of carbamyl phosphate synthase, it forms carbamyl phosphate. This carbamyl phosphate in the presence of aspartate transcarbamylase, it forms carbamyl aspartate. This carbamyl aspartate in the presence of dihydroorotase, it forms dihydroorotate. This dihydroorotate in the presence of dihydroorotate dehydrogenase, it forms orotate. This orotate in the presence of orotate phosphoribosyl transferase, it forms orotidine monophosphate. This orotidine monophosphate in the presence of the enzyme orotidine monophosphate decarboxylase, it forms uridine monophosphate. This uridine monophosphate in the presence of the enzyme kinase, it forms uridine diphosphate. Uridine diphosphate in the presence of ribonucleotide reductase, it forms DUDP. DUDP forms DUMP, which again in the presence of thymidylate synthase, it forms DTMP. This uridine monophosphate in the presence of kinase, it forms UDP, that is uridine diphosphate. This uridine diphosphate in the presence of kinase, it forms uridine triphosphate. Uridine triphosphate combines with glutamine, and in the presence of CTP synthase, it forms cytidine triphosphate. So let us incorporate the enzymes in between. Carbon dioxide combines with glutamine, and during this process 2 ATPs will form 2 ADPs, and in the presence of the enzyme carbamyl phosphate synthetase 2, it forms carbamyl phosphate. This carbamyl phosphate in the presence of aspartate transcarbamylase, which is a rate limiting step, it forms carbamyl aspartate. During this process, aspartate is added. This carbamyl aspartate in the presence of dihydroorotase, it forms dihydroorotate. Now this dihydroorotate will enter the mitochondria. This dihydroorotate will enter the mitochondria. Okay, and in the mitochondria, dihydroorotate in the presence of dihydroorotate dehydrogenase, it forms orotate, and during this process NAD gets converted to NADH plus H plus. Now this orotate in the presence of the enzyme orotate phosphoribosyl transferase, it forms orotidine monophosphate, and during this process PRPP gets converted to inorganic phosphate. Now this orotidine monophosphate, right, now this orotidine monophosphate in the presence of the enzyme orotidine monophosphate decarboxylase, it forms uridine monophosphate, and during this process carbon dioxide is removed. This uridine monophosphate in the presence of the kinase and along with ATP, where ATP gets converted to ADP, it forms uridine monophosphate. Now this uridine monophosphate in the presence of ribonucleotide reductase, it forms deoxyuridine diphosphate. This uridine diphosphate, deoxyuridine diphosphate again forms deoxyuridine monophosphate, which will again form deoxythymidine monophosphate. This uridine diphosphate gets converted to uridine monophosphate with the help of the enzyme CTP synthase. And during this process, sorry, uh, sorry, sorry, this uridine diphosphate gets converted to uridine monophosphate in the presence of the enzyme kinase. Because it is a kinase, it also requires ATP. So during this process, ATP gets converted to ADP. This uridine triphosphate in the presence of glutamine, it gives a CTP, and here the enzyme is CTP synthase. So this is the whole pathway of pyrimidine synthesis. Even if you do not remember anything, remember that the whole process of pyrimidine synthesis occurs in cytoplasm except this step which occurs in mitochondria. Then if you see the regulation of this pyrimidine synthesis, we have this here. The rate limiting enzyme of this pathway is aspartate transcarbamylase. This is the rate limiting enzyme, and this carbamyl phosphate synthase 2 is important because the enzyme PRPP will activate this carbamyl phosphate synthase 2, whereas uridine triphosphate will inhibit this carbamyl phosphate synthase 2. Okay, now if you remember, we have two carbamyl phosphate synthase. This carbamyl phosphate synthase is of two types. We have carbamyl phosphate synthase 2 and carbamyl phosphate synthase 1. If you see, this carbamyl phosphate synthase 1 is in urea cycle, whereas carbamyl phosphate synthase 2, obviously it is in pyrimidine synthesis. The source of carbamyl phosphate synthase 1 is ammonium ion in urea cycle. The source of carbamyl phosphate synthase 2 is amide nitrogen of glutamate. Then this carbamyl phosphate synthase 1 acts in mitochondria and it is present in mitochondria, whereas carbamyl phosphate synthase 2 is present in cytoplasm. Then this carbamyl phosphate synthase 1 is activated by the enzyme N-acetyl glutamate, whereas carbamyl phosphate synthase 2 is activated by the enzyme phosphoribosyl pyrophosphate and inhibited by uridine triphosphate. So this is important about the synthesis of pyrimidines. Okay, now if you see in the pyrimidine synthesis, if there is the deficiency of the enzyme, there is one deficiency of the enzyme which might result in a disease, that is if there is deficiency of the enzyme orotate phosphoribosyl transferase. The deficiency of the enzyme orotate phosphoribosyl transferase, this will result in orotic aciduria type 1, whereas deficiency of the enzyme orotidylate decarboxylase, this one, it will result in orotic aciduria type 2. Okay, so this orotic aciduria is a rare autosomal recessive condition and it is the most common error which occurs in pyrimidine synthesis. This OMP decarboxylase also is orotate type 1. I'm really sorry. Orotic aciduria type 1 occurs due to orotate phosphoribosyl transferase and OMP decarboxylase, whereas orotic aciduria type 2, it occurs due to orotidylate decarboxylase. Type 2 occurs due to orotidylate decarboxylase. Okay, so in this orotic aciduria, because of the deficiency of the enzymes like orotate phosphoribosyl transferase and OMP decarboxylase, there is increased orotate levels in the blood. Right? So because of this is responsible for the production of this CTP, UTP, CTP. For production of all these, we require this enzyme. But this pathway to work properly, but because there is no continuation of this pathway or decreased synthesis of these nucleotides, there is decreased CNS issues, like there will be growth retardation, developmental retardation will be there. This can result in megaloblastic anemia, and you will see increased levels of orotic acid or orotate levels in urine. The treatment of this disorder is to provide the end product. So obviously you are going to provide uridine derivatives for this disease. Okay, so this is important points about orotic aciduria. One more important thing is this orotic aciduria, which we are telling, this is mainly precipitated by allopurinol, 6-azaluridine. These are the enzymes which will actually precipitate this orotic aciduria. Right? So this is about the orotic acid, orotic aciduria. So thank you guys for watching my lecture. Thank you.
Thank you and thank you for watching.